me. But if anyone's ever worked with nematodes, they're not particularly easy to work with. They're super tiny. The taxonomy is extremely difficult. So after my PhD, I decided to go more into genomics because that was more my specialty. Working more with computational biology tools and high-frequency technologies to try and speed up the way we look at these small organisms that are otherwise pretty continually neglected. But I'm going to talk about some of this work that we do in my lab, mainly using marine sediments as a unifying theme. And then in the way that we set up projects in the lab is basically the interface between field biology and the more computational heavy sciences. So personally, I see myself as a translator who literally sits in the room between computer scientists and someone who goes out on a boat and translates between the two disciplines. So really trying to understand what's needed for field research, what are the questions and hypotheses in marine environments, and then also how can we develop computational tools and statistical approaches and data visualization to really maximize the amount of information we're getting from these newer high-frequency technologies. So, I have to know how to talk about code, but I also have to know that design a field study, which can be interesting from a career perspective. But my main central theme that I think about a lot is marine sediments. So marine sediments are really difficult to work with. We don't know much about the biodiversity of organisms that live in sediments, but they cover pretty much 70% of the Earth's surface. So wherever there's ocean, there's some sediment underneath that ocean. It's much harder to sample than surface waters. So typically for marine microbial ecology, you'll see a lot of studies where they're just going on boats and they're sampling in the water column and at the surface, and that's a lot of the high-frequency sequencing that you'll see in the field. Sediments, it's a lot, we're lagging behind, I think, in terms of molecular techniques, and that's mainly because of the difficulty in sampling. I mean, I work in the deep sea, so you have to send down thousands of meters of cable to go get sediments from some of the places and the sample sites that we work with. So we have a much poorer understanding of the biodiversity of the microbes that live in these sediments just because it's so difficult to actually get samples. But sediments themselves are a really great case study for ecology and evolutionary biology because there is, in theory, no biogeographic barriers that's separating the microbes that live here from the microbes that live here. There's probably a lot of factors that prevent dispersal, and there's probably a lot of microhabitat heterogeneity that we have to think about. But if you just look at a picture of sediments, there's no obvious biogeographic barriers like mountains in most of the sample sites that we work with. So, given that, we have this really high biodiversity marine systems, particularly for small microbial eukaryotes like nematodes, where you have this remote, extreme environment that's very food limited. So we're reliant on surface flux, anything that sinks. But you have this high species diversity. So this is kind of the paradox that we're dealing with, where we don't quite know why there's so many nematode species that live, and other species, protists, microbial eukaryotes, bacteria. We don't quite know why there's so many species in these remote environments. And my main driving question when designing studies and testing hypotheses is really figuring out the evolutionary ecological mechanisms which might explain high nematode biodiversity. Everyone's probably heard of C. elegans, right? C. elegans is a pretty boring nematode when it comes to taxonomy. It looks like nothing, just a worm, those pointy tail ends. And as an advocate of nematodes, I want to remind you that C. elegans is not a typical nematode. And actually, the reason why we use it as a model in the lab is because it's a really weird nematode that we can freeze and unfreeze. And it grows on plates, and it's happy doing that. You can't do that with most nematode species that actually live in the environment. And the species that we find in marine systems are actually really diverse in terms of their body organization. So we have all this incredible morphology. We have different tail shapes. We have these patterns on the outside of the nematode cuticle that can be dotted or striated. We have teeth, like giant teeth in some species. We have nematodes that are short and hairy and long and thin. So there's this incredible taxonomic diversity out there in these marine systems. And it makes for some really interesting case studies for ecology and evolution. To some extent, we can actually control the types of morphology that we're looking for. So we can look for nematodes body shapes in the environment, and we can design studies that basically use morphology as a variable that we control as part of the experiment. So it's not easy, and it's hard to pick these little guys out of your segment samples, but I think the hard work is worth it because of the questions that we can ask using these using nematodes. So the tree of life of nematodes is still pretty poorly known. We are still in the days of single gene phylogeny, unfortunately, for nematodes. We're still building our molecular frameworks primarily with eDNA ribosomal RNA, which is the gene that's historically been used for a lot of early phylogeny work. So although we do have some genomes, they're really not well distributed across the tree. So we have a lot of molecular work happening up in the crown clades of the tree of life. So C. elegans is basically all the way up in a more recently arisen clade, faster evolutionary rates up there. And the soil nematodes are really the clade where C. elegans is located. But the species that my lab focuses on is more early branching and divergent from C. elegans. So free-living marine species are much more early branching towards the base of the tree. We have a lot of morphological diversity. We have a lot of very deeply divergent lineages that inhabit marine sediments. And unfortunately for me, we don't have any genome data for any marine species. So all existing published genome data for nematodes is either derived from C. elegans, species that are closely related to C. elegans, which are free-living, or parasitic nematode species. So dog, heartworm, ascaris, onchocerciasis. There's a lot of tropical parasites that are nematodes and also sort of livestock parasites. And that's where the majority of research efforts really focuses on. So the marine species, the free-living species in the nematode tree of life tend to be pretty systematically ignored, apart from a couple of research groups, my group. So what we're trying to do now in the lab is trying to link molecules with morphology very strongly. So we can understand the morphological evolution of the biome, and we can get an idea of the drivers of diversity in the two. And we do this in two different ways, and I'll talk about both in the context of this talk. But I like to partition my thinking into big data and small data. So in the big data side of the lab, we're trying to use high-throughput sequencing technologies to basically rapidly sequence mud and the organisms that live in the mud to assess biodiversity and biogeographic patterns on a regional and a global basis. And this is your typical microbial ecology survey where you just have hard drives worth of data. You're not really looking at the organisms, but you're just trying to pull out patterns statistically. And then on the second hand, if you work with big data surveys, they can be quite frustrating because the data is so messy and sometimes it's unsatisfying. You can't quite test some hypotheses in the way that you might want to. So since starting my lab at Riverside, I've also kind of been obsessed with the idea of what if we could make things better by developing small data approaches. And by small data, I don't mean the data sets are small. They're still pretty big. It's still limited data sets, but I mean small in terms of artisanal, handcrafted, batch data, where we're physically trying to reduce the complexity of our genomic data sets or environmental data sets by really trying to do the hard work of taxonomy. So we're actually going back to taxonomic methods, picking out single nematodes from our samples, and we're carrying out sequencing on those individual worms. So we know exactly what went into a tube, which is in stark contrast to the big data approaches where you're just sequencing everything that's in that handful of run. Both have their benefits and drawbacks, and I'll talk about both in the context of this talk. But I'm going to focus first on the small data because this is what we've really been pushing out of the lab as the first kind of exciting. Well, I'm just super excited about developing these single worm sequencing approaches. So I'm going to talk about that in the context of some recent work we've been doing focused on nematode microbiomes. So the reason why you want to put a worm into a tube, I mean, not only for molecular phylogenetics, but sequencing technology is so good nowadays that we can actually profile the microbiomes of individual worms. And no one really knows anything about the microbiome of marine nematodes because no one really works on marine nematodes. But there has been a push in the C. elegans community, at least, to do some initial studies on microbiome profiles in C. elegans. And the emerging community is that it's actually really important for roundworms. Just like in most other phyla, I think we're understanding now that the microbiome is important potentially for the biodiversity and the ecology of the species. So one of the things we're thinking about is maybe the microbiome can help us understand the biodiversity a little bit better. And I just put some nematode stats there. So we have this high nematode species diversity in any given sediment. We have between 100,000 to 84 million nematodes per meter squared. We have 3,200 genera. We have hundreds of local species. And as we're going and looking at some of these species in sediments, we're finding microbiome associations, which are really exciting and compelling. So, for example, Laxus is SEN at the top. This is a stilbottom nematode that lives in tropical seagrass sediments. So coarse sandy sediments, and it has these sulfur-utilizing bacteria that are basically like a shaggy carpet on the outside of the body. And the nematode physically moves up and down in the anoxic and basically up and down through the redox layer of the sediments. And so the microbes are alternately exposed to oxygen and anoxic conditions. And in the sulfur cycling, that basically enables them to carry out their metabolism of all parts of the cycle. And then in other taxonomy, we're finding some potential microbiome associations inside nematodes. And this is just a very small example, but basically wherever we seem to be looking, as well as in C. elegans, we're finding what seems to be these conserved microbiome associations with nematode worms. But we've only looked at a certain number of species. So in my lab, one of the first studies we decided to focus on was to see if we could profile this more broadly and whether or not we could look at not just one species, but a variety of marine nematode species and ask whether or not the microbiome might play a role in the, or it might be associated with some of the morphological patterns that we're seeing in these nematodes. So one argument that you could make is that the microbiome would be an indicator of what nematodes are potentially feeding on in marine environments. And because there's so many nematodes, maybe the microbiome could actually give us an idea of selective feeding or niche partitioning or some sort of metabolic association with certain microbes. And then just to take a step backwards, I want to, at this point, give you a little primer on nematode taxonomy. So the reason why we're asking this question is because of the way that we've historically grouped things within this phylum. So back in 1953, a German taxonomist decided to look at a bunch of nematodes and group them according to their mouth parts. And this was done before the era of DNA, before we really had a good understanding of the molecular patterns in nematodes. So we have no idea if it's actually ecologically relevant or if it's just us humans trying to classify things. And so nematodes are grouped into 4 different feeding groups. These are thought to basically tell us a lot about what the ecology of these worms are. So we have group 1 and group 2. Basically, the feeding groups are determined based on whether or not the nematodes have teeth and how big their mouths are. So we have group 1A, which have very tiny mouths, really not much of an opening. And these are thought to be bacterial feeding nematodes that are very selectively feeding on specific strains of bacteria. Then we have group 1B nematodes, which are thought to be non-selective deposit feeders. So these are functioning more like sea cucumbers, where they're just shoveling up particles of sediment and kind of scraping off the biofilms or digesting the biofilms from the sediment grains. Then we have epistrate feeders, the 2A nematodes, which are, this is where we're starting to get lots of teeth in the mouth. So these have little denticles and rows of scraping-like teeth in their mouths. And these are more found in rocky endopetalons that kind of scrape algae and biofilms off larger pebbles and rocks. And then the group 2B nematodes are physically the largest nematodes, so the body size has to be quite massive. This is a group I did my PhD on. And these are carnivore scavengers, predators, and these can actually eat other nematodes, so you can find a nematode, half pulled apart nematode, which is pretty cool to see in the microscope. So when we look at nematode taxonomy, typically we would assign a feeding group to one of these nematodes. So you'd have a genus and you'd have a feeding group associated with that genus. And no one's ever really questioned whether or not, a nematode that has teeth is actually always a carnivore, or whether non-selective deposit feeders can actually eat things besides sand. So we decided to test this using microbiome sequencing. And what we were trying to figure out was, we were not sure what we were going to find. This was a preliminary study, but initially the idea was to see if we could detect coevolution between hosts and microbiomes, or at least detect some sort of selective feeding and niche partitioning, which might be determined based on these feeding groups. So we loosely define microbiome as any species found in association with that host organism. This is a very loose definition because we can't micro-dissect the nematodes. We don't know if they're going to have symbionts, but we know we can sequence profiles from single worms, so we can at least do a first-pass study. So this might include symbionts for some species. It would also include species found in host-associated habitat, so any bacteria that's stuck to mucus secretions or within the gut, the cuticle, which is the outer cover of the nematode. This would be either transient or resident taxa. So the purposes of this initial study, we didn't really differentiate. So that would include prey items and partially digested gut contents, obviously, and then any other types of human-species interactions that we can find. One of the things I'll show downstream is, this interesting ability of this data to actually show us potential parasitic associations, which I thought was really neat. And because nematodes occur everywhere in the world, we decided to design a study that was essentially just based on samples that I had in the freezer initially when I started my lab. And we selected a subset of sample sites which would allow us to test some different types of hypotheses. So what we ended up focusing on was a set of samples from off the coast of California, Gulf of Mexico, and the Arctic. And these were chosen because of the diverse geographic regions, but also because we had a mixture of different habitat types. So we had some depth transects up in the Arctic. We had an intertidal site from Laguna Beach that we just went down to examples. And then we had some chemosynthetic habitats where, so whale falls and methane seep habitats in the Gulf of Mexico. So we could test whether or not microbiomes were different in normal background sediments versus known chemosynthetic habitats where you might have specific symbiotic associations or specific metabolisms happening. And what we ended up doing from these samples is looking at the microbial assemblages associated with single worms. So here's where the hard part came in. So for taxonomy, I think you can kind of see here. So this is my thumb, and this is a pick that we use to pick nematodes out from our sediment sample. So it's either a fine tungsten wire or it's an eyelash mounted on a stick. And you have to look at that under the microscope. You have to hook the worm on that wire, on that stick, and then you have to, not shake, and transfer it over to a slide, mount that on the slide, put it on the microscope, and then generate your images of the worm. So this is why no one does nematode taxonomy. It's time consuming. This is why I want to teach them. But for the purposes of this study, it was really useful because no one usually does this. It would give us some relevant information about microbiomes and could link a microbiome profile back to a specific morphology. So we ended up getting morphological identifications and video capture images from single worms. We ended up with 281 worms that worked in the sequencing, representing 33 distinct morphological genera from 60 different sediment cores. So we were pretty happy with that as a first pass preliminary study. There were some worms that didn't work, but that data set was pretty robust. And then each worm went into a tube, and then we did a DNA extraction from a single worm. And from each worm in the tube, we generated 2 different metabar coding data sets. So we used the Earth microbiome primer sets, the B4 region 16S RNA to look at bacteria and archaeal profiles. And then we also did eukaryote metabar coding using the B1, B2 region 18S ribosome RNA. And so this was a really quick way to get us the molecular data from the nematodes. So we're basically barcoding the nematodes as well as any other eukaryotes that might have entered the gut. Linking the morphology with the molecular data in nematodes actually is really important. And then each of those were sequenced separately on mice. So for the nematode barcoding, the reason why this is important is because you have a lot of cryptic speciation in nematodes. You have a lot of convergent evolution of the morphology. So just because 2 worms look the same, they're probably not the same species. And oftentimes when we generate molecular data, and then when we go back to the morphological data, we'll find cryptic species that we can notice very commonly. And we see a lot of phylogenetic structure. And also geography doesn't tend to matter for nematodes. So it's very common This is a clade where we essentially have no sequence divergence, but we have nematodes from the Gulf of Mexico and the Arctic in the same clade. We have no idea why this pattern exists, but it's very, very common in marine nematodes. Just geography doesn't matter, and you have basically clades with global representation. So this is just a phylogeny that we built using the OTU sequences from our 18S metabarcoding from single worms. So essentially we looked through our data set and we basically chose the top OTU, the operational taxonomic unit from the 18S data, which was consistent with the morphology that we had identified from the individual worms. And then we just built a tree to look at clade structure and see if we could test for any microbiome patterns in a phylogenetic manner. And this was just a checkpoint too on the taxonomy to make sure that we were identifying things correctly, because sometimes we don't. And at this point, I just want to talk about a little bit about contamination, because this was something we were constantly paranoid with. And this is something that if you work with larger organisms, you probably don't have to think about a lot. But when you're dealing with individual worms in these tubes, you're talking about a worm that's maybe 1/10 of a millimeter to a millimeter in length. And you don't have much biomass to work with. When you're sequencing or when you're doing PCR amplifications on such a low amount of DNA, contamination is a constant thing that we're having to battle against. So we were worried about contamination from airborne dust, human skin, the kit microbiome, and usually for contamination, the lower biomass you have, the less DNA you have, the more signal of the contaminants that will come in. And this has been pretty consistently shown in a lot of the kit microbiome papers that have come out in recent years. So we knew about this, and we wanted to kind of head this off and make sure we're taking all sorts of controls to detect this in our data set. So what we did is we had really strict lab protocols. We tried to minimize contamination as much as possible. We had a PCR hood where we did everything, sterile equipment all around, gloves, molecular grade alcohol, throw things out if we're worried about contamination, just being pretty rigorous in the lab. But then in addition to that, we had this sequencing strategy, which was meant to capture all potential sources of contamination. So there's the prevention and then the detection. So we had a pretty rigorous set of blank and control samples that we sequenced alongside single worms. We did negative PCR controls. We included a positive PCR control as a mock community. This was a Zymo microbionics kit was the one that we used. It's just a kind of a mixture of DNA extraction from different species that you can find. And then we also had what we call taxonomy blanks in the study. So this is where we pretended to do taxonomy without actually keeping the worms. So my postdoc Tiago sat at the microscope and cleaned his pick, dipped it in the buffer, dipped it in the tube, dipped it on the slide, and then dipped it into the DNA extraction. We sequenced that too. So it was just meant to capture human-introduced contamination that might be introduced when we're processing these worms. And then we sequenced everything alongside our actual data sets. And then when we got to the data analysis side of things, we used like really stringent biomimetic filtering. So basically, if an OTU was found in the contaminant samples, we completely subtracted that OTU from the data set. So this is probably overly rigorous and overly cautious, but we just wanted to make sure that our microbiome samples were actually representative of what was in the nematode gut. And the reviewers seemed to be pretty happy with that, so that made us happy. The reason why we did that is because when we initially looked at the data set with the blank samples in, we saw this kind of pattern. So our positive PCR controls were always really separate from our data, which was good because they're a very different community. And for the most part, the control samples, the blank samples, wouldn't have enough reads, so they would drop out during the rarefaction. But occasionally, a couple samples would actually have quite a high sequencing depth. So this blank sample, that blue sample there, looks just like the microbiome samples, which is why we decided to filter out all these OTUs, because we didn't want that confounding our analysis. All right, so now to the actual results. So after we did that filtering, after we dealt with contamination, after we checked our nematode phylogeny, then we decided to look at the microbiome profiles and see whether or not we could find any patterns, essentially, according to these feeding groups and different variables. The first result is that we had some sequencing data available from the raw sediments. So this was just bulk sediment that was sequenced with CCNS. And the sediments themselves are very separate and distinct from the nematode microbiomes. So nematode microbiomes tend to be much lower diversity in their bacterial community. The sediments tend to be more complex and just have more OTUs. And this is consistent with studies of C. elegans and other soil nematodes. So you always see this nematode microbiome profile being much lower diversity than the actual substrate that the worm is living in. So that was good. That was reassuring. However, when we went and looked at the nematode microbiome data, this is where we got frustrated because basically the spoiler of the study is that we found nothing. We found no patterns. Which was, it could have been difficult to publish, but we were actually included in a special issue of molecular ecology. So that was, I'm really glad we got this study out there because I think the lack of patterns is even more mind-blowing than if we had found patterns. So the feeding groups are labeled by blue and green. So we have our group 1 in blue, group 2 in green, and then we have ocean regions according to the symbol. And essentially, there's no clustering of the microbiomes according to nematode feeding groups. So this idea that all nematodes with small mouths are eating specific bacterial strains doesn't seem to hold up. Because if you look in the PCOA, the microbiomes can be pretty similar to predatory nematodes. And we did this in multiple ways. We colored it by ocean region, by habitat type, holds methane, chemosynthetic versus background sediment. Nothing we seem to do gave us any sort of pattern. We always just got these really frustrating PCOAs where we just had nothing kind of coming out. And these are unifrac analyses, unbelievable unifrac analyses over time. We also redid these analyses with ASVs, ample sequence variants, instead of OTUs. Same thing. So the bioinformatics doesn't seem to change this lack of patterns, even where we use essentially 100% OTUs. Looking at it in a different way, we tried to look at, well, maybe nematode taxonomy at the genus or family level would lead to some clustering, even if the feeding groups weren't relevant. And looking at that in a different way, the families or the symbols now, we're still not getting any sort of clustering by nematode taxonomy. And this is a 60S data still. For the eukaryotic microbiome, we're still seeing nothing. I mean, I would say with the eukaryotic data, the diversity of the eukaryotic microbiome was less. So a lot of samples dropped out of the eukaryotic analysis. And you can kind of squint your eyes and say maybe you can make an argument for some 2D nematodes clustering out separately. But I think this might actually be reflective of sequence artifacts or read depth and not maybe reflective of true patterns. So we subtracted all the nematode OTUs from this, so this is just the protists and non-menzone OTUs in the microbiome, like diatoms and algae and things like that. So we saw this similar lack of pattern across multiple data sets, 18S and 16S, which was reassuring, but again, frustrating. And this is the paper, so this is published in Molecular Ecology, and that was our title, literally. There was no pattern. If you look at the profiles in terms of microbial taxonomy, this is an image representing essentially one genus of nematodes, so species where the morphology is the same. And it doesn't really matter. If you're looking at the same species, the microbiome profile seems to be pretty stochastic. So, when we were looking at these data, it was very clear that just because you had two species from the same site doesn't mean their microbiome profile is going to be similar to each other. It could be very, very different from one another. And the dominant group in different nematodes seem to vary quite a bit. So that's kind of the data that we put in that initial paper. And I think having worked with the data set, we said there must be more to the story. So we looked initially at the overview patterns to see if we could see anything obvious. But now we're working with these data sets because they're so hard won and the taxonomy is so difficult. I think we're trying to dig into these data a little bit more and find stories and see if we can find any specific taxa or specific associations which might be associated with specific nematodes at certain sites or in certain taxonomic groups. So what we're doing now is we're building these. These are figures from a package called Metacoder. So these are essentially visualizations of the taxonomic hierarchy. And each of these little graphs is of the microbiome of an individual worm. And you can see that the microbiome profiles are pretty different. So we have some that are sparse, some that are bushier, have more taxa. And we're trying to just look through these hierarchies and look through the actual bacterial taxonomy to see if the taxonomy is informative. And you'll see there's a little archaea in there too. The little, there we go, there's an archaea. So we do find archaea as well bacteria associated with the worms, but the bacteria seem to be more dominant in the microbiome. So we're looking at the taxonomy and we're trying to, come up with some hypotheses, or we're at least trying to identify some bacterial taxa which might be important in nematode feeding preferences or maintain specific association with nematode worms. The reassuring thing about these data sets is that in fact, some of the bacterial taxa we're finding are indicative of marine habitat. So this is, in addition to the contaminant filtering, it's kind of reassuring to see the bacterial groups that are that we would expect to find from our sample sites. So we have polar bacteria, we have benthic bacteria, and typical marine Gamma-proteobacteria and bacteria. But we also find some kind of intriguing taxa from a nutrient cycling perspective, and this is something we're digging into further. And we're actually in the process of generating metagenome data from these individual worms to try and circumvent some of the issues with doing taxonomy from 16S data. But we find in the nematode microbiome these taxa which are pretty characteristic of nitrogen, methane, and sulfur cycling. So we have ammonia oxidizing bacteria and archaea, we have methanogenic, methanotrophic taxa, sulfate-reducing bacteria, and we also have found some vegetative, which are mat-forming bacteria at the methane sinks. And so I think this is really interesting because if nematodes are either feeding on these nutrient cycling bacteria or maintaining some sort of association, it suggests that they're doing a lot in the environment that we should probably know about, we should probably budget for in our global models of these nutrients. So we think a lot about methane and carbon cycling. If nematodes are eating the bacteria, I mean, this is a big part of the food web that we're really missing out on right now. And this is just an example of kind of a more interesting nematode microbiome from one of the cold seed methane seed sites. And so we find the nematodes eating potentially this dominant mat-forming bacteria at these sites. And then we also found some bacteria which were consistent with some of the results of C. elegans. So the Acetobacter was actually reported from some of the soil nematode microbiome studies, which I think is interesting because that might suggest some evolutionary conservation between certain nematodes and certain bacterial groups across all environments, which would be fascinating if true. And so what we're doing right now is we're generating more figures to dig into the bacterial taxonomy more, but we're also using the 16S data to generate mock metagenomes from these communities. So we're using PyCrust2 to look at predicted tags, which might be in these bacterial genomes, just to get more out of these data sets and to try and generate some more hypotheses. And then alongside that, we're generating actual metagenomes. So we're looking at the actual gene content to see if we can detect some of these nitrogen cycling, methane cycling genes actually present in the nematode microbiome. The other neat thing that we found is potentially parasitic interactions in nematodes. So if you're seeing them in the microbiome, you're getting parasites all over the place. We found this list of, I'm just Googling taxonomy of the microbiome taxa, and there was some indication that there were these divergent weird parasites that were found in the nematode microbiomes. So we don't know if these are parasitizing nematodes or if these are parasites of the food sources of nematodes or just in the sediments. But I think that this idea of single worm sequencing we can use to ask these really broad questions. So other types of ecological interactions besides just feeding, like parasitism. I think this approach of this small data is really got me hooked in terms of the amount of information and the resolution of the information that we can get. So we're moving forward on this. Like I said, the takeaway is that for the moment, at least from the 18S and 16S data sets, we don't see any patterns. We're trying to figure out what this means. One hypothesis is that nematodes are actually just pretty generally feeding on things, or they don't have any obvious microbiome profiles separating different feeding groups, different species. Or it could be a limitation of the study design. So I actually personally think it's probably the latter. One of the emerging views from the microbial ecology literature is that species don't matter as much as genes, and as long as communities are conserved in terms of the functional genes which are present, it doesn't really matter what taxa are there. So this is one of the rationales for generating metagenome sequencing. Is I think we really need to look at the functional genes in these bacterial communities, and we might actually see some clustering based on the metagenome. So maybe nematodes in group 1A are always feeding on specific bacteria that produce specific metabolites. So, maybe they're going after something that smells good in the environment. We think that nematodes do a lot based on chemosensing. They have a lot of glands and organs that are designed for detecting chemicals in the environment. So it wouldn't surprise me if they actually have evolved to go for bacteria with certain chemicals, irrelevant of the 16S gene that bacteria might have. And then the other thing that we haven't really captured with the sequencing approach is what is this microbiome? Is it transient? Is it resident? Is it active versus inactive? So my graduate student, Patricia, is working on all these questions. And one of the things that she's going to be doing in the next couple months is trying to develop these fluorescence in situ hybridization techniques to look at where the bacteria are actually situated in nematodes, if it's in the gut. Try to get some biomass estimates and try and look at visualizations of different specific taxa based on the 16S data. So that's a lot of work. But I'm going to zoom out at this point and talk about shifting gears towards the other type of work that's going on in the lab, towards the big data approaches. And I would say that these two approaches kind of inform each other, so we can, we're probably at some point going to integrate nematode microbiomes into the bigger data studies, but I just want to talk about if you're not doing the hard work of taxonomy, if you're deciding to sequence everything, you're focusing on mud more generally, what can you do with those surveys? So, it is pretty fast to get sequencing nowadays in microbial ecology, and I think in marine systems we still have a lot of questions which they've been addressed fairly well at this point in terrestrial systems, but not so much in marine ecosystems. So I think for the bigger big DNA or big data approaches, we're trying to take what's been done in terrestrial environments and apply it to marine environments. So one of the main questions is basically this idea of, the vast Beckman's hypothesis, everything's everywhere, the market selects. So do we actually find a taxon globally distributed, or do we actually see some regional phylogeography and restriction of taxa? If we're not, and specifically in marine sediments, I would say. Secondly, in marine sediments, we don't really know what determines microbial community assembly. So we will want to think about the abiotic conditions as well as the biological interactions that will result in a community at any given place and time. And this is sort of being done now with sequencing on a taxonomically broad scale. Previously, we asked this question for specific organisms, specific phyla, but now with sequencing, we can really assess that in terms of the whole community. Thirdly, this idea that genes maybe matter more than species. I really think this is the future of microbial ecology. This is genes, not 16S data. And then finally, this idea of, okay, well, we're assessing all these hypotheses, but that doesn't really help us manage these ecosystems or respond to climate change. And the last facet of what we're trying to do with big data studies is really using data-driven science to not only predict habitat characteristics, but also develop some measures of ecosystem health using these eDNA sequencing approaches. Not only just looking at biodiversity, but also looking at gene pathways and whether or not we can use these in environmental management context. So for these types of studies, we're using a pretty similar workflow, I would say, to the nematode microbiome studies. And all microbial ecology studies are essentially doing the same thing. So we're collecting samples, we're extracting DNA, we're either doing PCR or not. We're doing something in the lab, and then we're sequencing, and then we're generating figures for publication eventually. That's the bottleneck. But I did want to highlight a specific modification that we use in our protocol, which is between sample collection and DNA extraction for the type of work that we do in the lab. If we're focused on metazoa and microbial eukaryotes, typically we need to isolate organisms physically before we go into the DNA extractions. And this is pretty distinct from what you would do in a normal microbial ecology study. So usually, if you read soil papers, they're just putting 2.5 grams of sediment into, or soil into a DNA extraction tube. We don't do that, so we use a decantation and sieving protocol to basically get rid of as much sediment as possible and to concentrate the nematodes and the other myofauna microbial eukaryotes down into a little tube. And this is so we can accurately capture the contaminant. So we're dealing with sediment volumes of maybe 500 mils or 100 grams of sediment. It varies a little bit based on the study design and how much we have available. But this is a big distinction. So if you read papers where they put 2.5 grams of soil into a tube and they said they didn't find much nematodes, that is why, because we have to do this to get the nematode data. But we're also washing away things. So we know we're washing away most of the protists. So to give you an idea of some of the surveys that we're doing in the lab at the moment, we're focusing on 2 regions, Alaska and Southern California, which I'll talk about next. And basically, these studies are characterized by a lot of samples. I would say probably not as many samples as some of the human microbiome studies at this point or the soil ecology studies, but we're trying as hard as we can to get hundreds of samples into these sequencing pipelines because we just want to broadly assess patterns across different regions and geographic areas. So it's harder to get marine sediment samples, so that's why our sample size is pretty smaller. But for this Arctic project, this is basically motivated on the fact that there's no genomic baseline data in the Arctic, despite the fact that we have climate change happening, sea ice cover is decreasing. All of the studies previously in these regions have been morphological or traditional. There's a lot of fisheries surveys, things like that, but there's no one's been sequencing anything, especially no one's definitely been sequencing nematodes. So, part of this rationale is just getting some genomic data to do some baseline characterization of the patterns that exist before climate change takes over. And for this study, we're focusing on a range of sample sites in the Beaufort and Chukchi Seas. So we're comparing diversity between the ocean regions and also looking at patterning within specific transects in specific different regions. And we're using the integrated mix of data, so we're doing metabark coding, so we're doing 18S and 16S metabark coding, nematode taxonomy for a subset of samples, so we're looking at the worms. And then we also do have a selection of sites we've selected for metagenomic sequencing. We're doing raw and processed sediments, so we're doing the traditional 2.5 grams of sediment in a tube, but then we're also processing the sediments to try and capture the myofauna community a little bit better. And then basically, we have a little bit of temporal information, but it's just a lot of data, a lot of environmental metadata that we're all trying to analyze together. And environmental metadata is basically any sort of context that we need for the sediment samples to try and figure out what the environmental drivers of the community structure are. So for this study, we have sediment cores, so we have our basic information from the ships, depth, salinity, temperature. But then from the sediment cores, we've basically been paying contract labs to generate nitrogen-15, carbon-13, chlorophyll A, phthalate, total organic carbon. And this just tells us a little bit about the habitat because we can't see the sediments. We don't really know what's happening there. But we can profile the sediments in such a way that we can look at the sources of nitrogen. We can look at degraded versus fresh marine snow in these environments. And we can look at just the total pool of organic carbon, things like that. And that can help us understand what factors might be associated with patterns in specific habitats. So we're still analyzing these data. So some preliminary results here are region tends to be important. So the Beaufort and the Chukchi Seas in the Arctic is very different. Water mass is very different hydrodynamic regimes. And unsurprisingly, we see that in terms of the sample separation. So the sites are very different. But when you get down to a regional level, the pattern gets a little more complicated. So only 10% of the variance ends up being attributed to region in a given area. This is the Beaufort Sea. And temporal factors don't seem to play a very important role. So we're still trying to make sense of this data. The picture is pretty complicated. Overall, depth seems to be an important driver of community structure. And this is reassuring because this is actually depth is a known differentiator of community structure in marine sediments. So the fact that we see depth as this major driver in the microbial studies is reassuring and not surprising and consistent with the historical literature. And then we're seeing in the bacterial data, some correlations in diversity between things like nitrogen 15 and salinity. So there's, a graduate student, Alexis Walker, is working on this project and is still in the weeds of data analysis for this data set at the moment. The other thing which we've been thinking about, because we've been seeing in the data set, is in the Arctic, we're actually detecting quite a lot of harmful algal blooms, so Alexandrian species. So these, the dinoflagellates, basically this big green part of the bar chart, seems to be pretty dominant in the Chukchi samples. And one of our hypotheses is this might be potentially representing benthic cysts. So Alexandrium can basically lie dormant in the sediments until conditions are right, and then it emerges, and then an algal bloom takes over. And this is important from an environmental managing perspective because you want to know if this is going to impact your fisheries. And seeing signatures of taxa that we don't necessarily expect to find, I think, is one of the more exciting things in these data sets. So we're trying to develop some hypotheses and come up with ways where we can test whether or not this is, is this really a benthic cyst that's in the sediment, or is this just a signature of DNA that's falling from the surface waters? Because you do typically also tend to see a pretty strong signature of phytoplankton and anything that breaks down from the marine ecosystem. So, sediments are good but also bad because they are sort of the ideal environment for preserving DNA long term. And you do see signatures of organisms which fall down from above, including fish and fish parasites, things like that. All right, and then we're doing a similar study in Southern California. So I would say the same methods are being applied to a more local study. And again, in the Southern California Bike, we are having hundreds of sediment samples. The difference with this other study compared to the Arctic is that the Southern California study is more focused on biomonitoring. So this is a project, another large collaborative project that's being carried out in conjunction with the Southern Coastal California Water Research Project to support down in Costa Mesa. And they've been great at reaching out to me at Riverside and setting me up with collaborators in the Southern California region. And this is a Southern California Bike Survey, if anyone's heard of it. This survey happens every 5 years, from Santa Barbara all the way down to Tijuana in Mexico, basically. And they've never done any sequencing, and they've never done any myofauna in these surveys. So they went out sampling last year, and we basically put up our hands and said, if you have a sediment course, we'll give you nematode data and sequencing data, which they were excited about. So we're doing the same approach as in the Arctic data set. We're generating this multi-marker metabarcoding study, possibly adding on some metagenomics. And then we have this extensive suite of environmental metadata, again, associated with the sediment cores. The difference in California is that we're more focused on environmental impacts. So we have the Port of LA, where we have some more pristine versus more impacted sites. We're actually, we're curious to see if we can see a signature of environmental disturbance and pollution in this data set. And then we're also interested if we can detect certain bioindicator taxa that are associated with the most polluted sites. So maybe there's a certain nematode that loves DBT, and if we can find that nematode, then we can use that as a proxy of places that we should avoid swimming in. And this is why the collaboration is really important, because it's actually really expensive to generate these profiles of the sediments. And the great thing about this collaboration is that because it's an ongoing survey, they've been doing it for 20 years or so, and they will collect sediment cores and calculate all of these measurements. So grain size, they'll do biogeochemistry, they'll give us measures of legacy pesticides, as well as emerging contaminants. My grad student, Hannah, who's working on this project, is particularly interested in microplastics and whether or not microplastics might impact nematode community structure. Nematodes tend to be controlled by sediment grain size. So, one interesting idea we have for microplastics is you have essentially little beads which are mimicking sediment, and that might change the properties of the sediment. So you might actually see changes in nematode community structure if you have areas that are really heavily polluted with microplastic beads. So, these projects, I would say, are less well developed at this point. I mean, in terms of we just have a lot of data we have to analyze sitting on hard drives. But generally in marine studies, with these big data studies, we do see depth being a major driver of microbial community structure, not just for bacteria and archaea, but also for things like nematodes. So what we're trying to do from these large data sets is identify microbial bioindicators of disturbance or different habitat types. And it just essentially involves sequencing a lot and trying to analyze these large data sets in conjunction with these environmental metadata characteristics. Eventually, we'd like to add on some nematode microbiome work there too. So one thing I'm particularly interested in is whether or not the polluted sites would influence the nematode microbiome, or you would get specific microbiome associations in these really heavily impacted sites. Definitely from the C. elegans literature, the microbiome is known to protect against environmental stressors and things like pathogens. So certain bacterial taxa can confer resistance in the nematode and result in no drop in fitness in the nematode. So exploring some of these patterns in an environmental context, I think, would be really important. So I'll start to finish up there. I'm just going to give a quick plug for the last little facet of my research, which is developing software tools. And so Felipe mentioned we work on data visualization. And one of the parallel projects we have to the hypothesis-driven research is trying to develop tools which are going to help us deal with these data sets in a way that's better than writing our code for 3 months. And this is a project which we're launching actually not next week, but the week after, which is, so Finch is a data visualization project I've worked on for several years, funded by the Alfred Foundation. And we have, through our second round of funding, we basically revamped this web portal, and we're re-releasing it as a standalone desktop app, which is, this is just a GIF of the visualization app. So it's basically like Slack for microbiome data, I guess you can think of it that way. And this is a way to take your big text file, your big OTU table that you get from a metabarcoding or a metagenomics data set, and just put it into a visual context so that you don't have to spend hours or days or weeks trying to write code to pull up patterns. So the idea is that visualization tools will just very rapidly help you look through your data set, send your data set to collaborators, and help you understand the patterns that might be present in that data set. And so you can use that to guide the next round of sequencing, or you can use that to start to formulate hypotheses from your sample sets and your questions. And so what you're seeing here, everything that's visualized is basically constructed on the fly from the text file that you upload into this framework. So it's detecting all of the metadata, all of the sample names, all of the proportions of the taxa. It's detecting the taxonomic hierarchies, and all the metadata characteristics. So, like I said, you can use this framework to upload metagenomics or metabarcoding data sets. We're having a people demonstration later. We don't quite have time to talk about all of it. But it's an open source project developed by a data visualization studio in Oakland. So I think the exciting thing about this project is it's not being developed by scientists, it's being developed by a studio who's experts in this. And so one of the things we're doing, I've been looking for potential signatures of microbial symbionts in some of our archive data sets, just using this little autocomplete search box up here to look at specific taxonomic names. But I would encourage you to go to the website. So it's finch.org, and we're releasing this desktop app on April 29th. And we will release it on that date because it's the industry studio that's doing it. So it's not scientists. They're very on time with things. And I'll have, like I said, I'm happy to give anyone a download link beforehand or give anyone a free trial. So I will just say, with that, I'll say thank you. So thank you for the invitation to talk here. And really enjoyed this meeting and I'll take any questions. Where to start?
You mentioned the temporal perspective. I'm just wondering if you'd actually try to do coring so that you go back kind of see if there's turnover. That would give you some insight on this hypothesis and maybe more over a long time period.
Yeah, that's something we thought about. And so they've definitely done that with the crocus. They've gone back and the province of DNA gets chopped up, so you have to modify your metabarcoding locus that you're looking at. But they can definitely go for forams, and they've done comparisons between sediment cores with microfossils and DNA. Because you get non-overlapping signatures, some taxons are better in one versus the other. But that's something we definitely thought about. I don't know for nematodes if it would be possible because they do vertically migrate up and down sediment cores, so it might be tough. But I mean, I think sediments just have this untapped potential for this historical insight.
I was wondering, so for the first kind of single nematodes that you didn't necessarily find patterns in the initial analysis, and for some nematodes, at least, and obviously not a lot of them done, but for the gut symbionts, they don't always strongly select in the gut, right? So you might expect this kind of large variation. But I was wondering if in any of your samples, you would expect, like, if you had those epibionts, that they would be kind of like the drastic majority of your sample, right?
Yeah.
So do you have any patterns where you see, not necessarily like that the whole community has patterns, but that whatever the dominant symbiont is has patterns to see if maybe you have patterns of like who has epibionts or who doesn't have?
Yeah. So with them, we know exactly what genera would have the epibionts, and we're actually looking for those genera in our sediments, and we couldn't find any. So I think our data set doesn't really have any of these characteristic epibionts, taxo, because we just would see that in the taxonomy, and even under the microscope, epibionts are very obvious under the light microscope. So yeah, you would expect those to be an outlier, and that's kind of what we were searching for, but we're at the mercy of the worms that we find in the sediment core, and we just couldn't find them. The ones with epibionts are actually pretty fragile, so they could be really long and thin, and I think they just get broken up a lot of times when we're processing the sediments.
I was relieved to see that the diversity of the microorganisms was different in the sediment sample versus the nematode. So is that, so that I understand the sediment sample was not, you didn't take out the nematodes, it was bulk sediment that you sequenced, right?
It was, so the sediments, the yellow triangles were bulk sediment, and then everything else was nematodes, microbiomes, so that was a wash nematode in the tube.
So that means, in other words, we don't have to worry, if we want to understand like how is the non-animal associated microbial community, how is that correlated to geochemical parameters? We don't have to worry about those that are in symbiosis or in the guts or whatever of the animals because they make such a minority.
I would say so, yeah, because you have such a high biomass of bacteria in just the bulk sediment. Even if you got a nematode in there or like a piece of it, I would say it would be pretty swamped out by the diversity of the microbes in the sediment. But you would assume you could also detect that signature, and the symbionts tend to be pretty specific, so you could also see if that's the case in the data.
So you mentioned looking for functional gene groups rather than actual genomes to try to pull out some distinctions in the data. I don't know if there's any. One thing that jumped out in my mind was EPS secreting versus non-EPS secreting. I would imagine EPS would be like copper-taking magnesium. So
Is that extracellular?
and like the extra polymeric substance, like the polysaccharides that some microbes, but I don't know if there's other things that you maybe have an eye on.
Yeah, I mean, specifically we're looking for, I would say, nitrogen cycling genes. So basically we're the stereotypical sulfur cycling, methane cycling, things like MOA and NIF-H, things like that, I think is what we're looking for, but definitely if you have other suggestions for the proteins, then we will look for them too. I think that's part of the metagenomes is hard because you kind of have to know what you're looking for in metagenomic data sets, and that's why it's so messy and frustrating oftentimes. And I don't personally have a bacterial, proper microbiology background, so I think it's hard for us to read the literature and figure out what's important. So that's why I like talking to people who have suggestions for what to look in the data sets.
Great, that's question.
Sorry, great talk. I'm just wondering about the Bike survey. You mentioned like there has been like 5 surveys before, but can you like still go back to like extract DNA from those surveys, or that's like already like those samples now? Like
They, I don't think they typically freeze the sediments. So what they typically they would collect and they would process for the different analytical chemistries and then also for macrofauna. So Although I have met a couple people who say they have stuff in the freezer, but I think that's just a chance occurrence. I think there's no, at least from my understanding, there's no long-term banking of frozen sediment cores because they just want to generate the data and everyone's freezer is full, basically.
Right, so let's thank Holly.